and I will introduce Dr. Jiwon Kim. She will be our first speaker today. She is a current postdoc in the lab of Dr. Ian Wong at Brown University. She received her PhD in mechanical engineering at the Korea Advanced Institute of Science and Technology in the Daejeon Republic of Korea. She has many first author and co-author publications, including one first author that's currently under review at Nature Physics. So good luck, and I hope your reviews go well. She has also received many outstanding paper awards through the KSME Bioengineering Division Spring Conference. So taken together, you are also a very impressive scientist, and I look forward to your talk, which is titled Programmable Transitions from Orbiting to Matrix Invasion in Epithelial Spheroids via Dynamic Physical Confinement. So with that, I will stop sharing my screen and allow you to go ahead and start your talk. Thank you so much.
Thank you very much for a kind introduction. And thank you, Kaya, Luria, Lani, every co-organizers for organizing this. I'll share my slide. So, can you see my slide in presentation mode?
Your screen is not showing at the moment.
Okay.
There we go.
Oh,
it's starting.
It's
Here you go.
coming.
Okay,
Perfect.
thank you. So thank you very much for the invitation. It's great to be here. I really appreciate it. Okay, I will start. My research focuses on spheroid dynamics in 3D, but along the way, I've come to realize that the intermediate filament have very important roles in the process. And today I will share two of my unpublished works. One is more about the orbiting spheroids and vimentin, and another is about cancer mechanical memory and keratin. So, okay, let's begin with the orbiting spheroid first. So here's a memory epithelial cell spheroids embedded in a collagen matrix, and now as time passes, they might migrate in circumferential directions, orbiting within the spheroid. So here you can see good examples of collective orbiting in the memory gland cell spheroids within a 3D matrix. So these rotations play a crucial role in shaping tissue morphology, in other words, the morphogenesis. Okay. And the sum of the cells may break away from the spheroid, migrating outward and embed into the surrounding matrices, like this one. So they can migrate in circumferential directions or radial directions. However, spheroid dynamics in 3D matrices are poorly understood because the cells and matrices are affecting each other. This is called the dynamic reciprocity. For example, the cells are attracted by fibrotic ECM because it is easier to grab and move along. However, as much as the 3D matrix can change the cell migration also remodels the 3D matrices, and this dynamic reciprocity results in emergent behavior, which makes it very difficult to study. So in our experimental setup, we try to visualize the cell migration and in the matrix deformation at the same time. So here, the cells expresses fluorescent signals in their nuclei, and we also added some fluorescent beads in the matrices so that we can reverse calculate the matrix deformation from the bead displacement. So here's what happens. So at first, the spheroid starts to rotate for a while. And if you look closely, the ECMs nearby the spheroid twitches along with it. And next, the cells begin to invade, grabbing and pulling the ECM as they move through this. We observed multiple spheroids, and then we identified that there are 3 distinct phases in their behaviors, which is pre-orbiting, orbiting, and invasion. So here, the below chiral graph shows the relative angular locations of the nuclei over time. And for the first 6 hours, the outermost cells shows minimal movement as seen as these parallel lines on the kymograph. But in the next 6 hours, the cells suddenly start to rotate around the spheroid. And now the kymograph shows a slope, which means that these cells are orbiting all together. And after the orbiting motion stops, we can see that the cells invade into the surrounding matrix with irregular patterns on the kymographs. So there are 3 phases. Then we extracted the vector fields from the time-lapse images, and we decomposed the velocities into the circumperiodic profiles and then the radial profile. So here the green to purple color scheme shows the circumperiodic velocity, and then the red and blue shows the radial velocity. And about like 9 hours after being embedded in the collagen gel, the most of the spheroid area appears in green color here, which means that the majority of the cells are orbiting all together in a very high speed over like 10 micrometer per hour. And over time, the green area become less and less dominant. But now, when you see the radial velocity, the prominent red regions appears at the top and the bottom of the spheroid, indicating that the cells are beginning to migrate outward of the spheroids in radial way. So we wanted to know how this collective transition was shaped by the local curvatures under the dynamic reciprocity. And unexpectedly, we observed that most of the spheroids were ellipsoidal rather than perfectly spherical. So they are like the aspect ratio is like 1.1, especially being after embedded in the collagen gel. So you can see this spheroid looks like an egg. Why this matter is that the spheroid, which were initially somewhat elongated, becomes dually spherical during the orbiting phase. And after the orbiting phase, the spheroid started to invade, and then the invading branches emerges at the top and the bottom of the spheroids, which is consistent with the initial major axis. So here the red means that this part is little protruding, more protruding than the average, and then blue means the opposite. And during orbiting, the roundness peaks and then goes down, leaving the branches here and here. So how comes this morphological consistency is possible? So when the spheroid gets rounded, it locally pulls the surrounding matrices to make it fit to the new curvature. And after that, now the cells in turn migrate toward the remodeled ECM, resulting in a morphological consistency we observed previously. So here we take a closer look at the tractions, and during the orbiting phase, the inward tractions, which shown as blue bars here, seems to be exclusively accumulates at the top and the bottom of the spheroids here as the spheroids gets rounded, and then the bias becomes more dramatic during the invasion like this. And so we took a closer look at the collagen structure near these spheroids, and this is time 0. So the spheroids are still ellipsoidal, and then the collagen structure seems very isotropic here. However, at time after 15 hours, the orbiting spheroids is over, orbiting phase is over, and the collagen structure seems very aligned near the spheroids, again confirming the locally concentrated matrix deformation. So to summarize so far, during the orbiting, the spheroids became more rounded, and to match the decreased curvature, the surrounding ECM gets remodeled in contractile directions, and this makes the collagen fiber alignment. And as a result, invasive strands emerge near the initially protruded regions, resulting in the morphological consistency. So next, we moved forward to explore the effect of cellular heterogeneity in the spheroid behavior. Here, the pink cells are engineered to induce EMT phenotype. We mixed in the snail cells, we call it snail cells at the ratio of 10, 30, and 100% to create these different kinds of mosaic spheroids. And compared to the wild-type cells, the induced pink cells were larger and more elongated, and they expressed more pimentin in them. So first, this is the 10% mosaic spheroids, and then they exhibited similar behaviors that were observed in the wild-type spheroids, where the spheroids start orbiting and then transition into collective invasion. And the slope here, slope in the kymograph shows that the cells are orbiting. And interestingly, those snail cells tend to be salted either at the core or at the periphery of the spheroids. And then we increase these snail cells up to 30%. And notably, there was no orbiting motion at all. So the velocity profile seems just very random. And the kymograph looks parallel. But still, the snail cells were solid like this at the periphery or at the core. And though they did not undergo any orbiting phases, they were very able to embed into the matrix. So when we see the traction patterns, the 10% mosaic spheroids looks similar to the wild-type spheroids. So the traction hotspots are coincides with the initial elongation axis, but in SNE 30% mosaic spheroids, the traction hotspots just coincided with the snail cells at these 3 points. And the collagen alignment were also significantly aligned only there at the snail cells like this. Thus, here we concluded that the snail cells can remodel the surrounding matrices without undergoing the specific collective orbiting phases. So they have different mode of migration. However, when it comes to 100% snail cells, which expresses pimentin a lot, both the orbiting and invasion were suppressed. So it just continued to grow larger over time without any notable migrations. Finally, we collaborated with Professor Carlos' group and developed a computation model to better understand the role of adhesive forces in this orbiting to invasion transition. And in this model, each cell is self-profiled and its migration is regulated by repulsive and attractive interactions, both between their neighboring cells or between the cell and then the ECM components. And this model could even capture the behaviors of mosaic spheroids. The collective orbiting behavior was reproduced with the 10% snail model here. But so you can see that the cells are orbiting coordinatedly, but in 30% snail, they did not orbit at all. The below graph, the angular momentum visualizes the extent of coordinated orbiting behaviors, and then you can see that 30% snail model failed to make the coordinated orbiting behaviors. Here, the red and blue lines indicate different input variables, and in both cases, with an increasing portion of the snail cells, we can see that the average angular momentum dramatically decreases like this, especially between 10% and 15% here. So yes, the model successfully simulated the spheroidal dynamics we observed from the experimental data. So to summarize the first chapter, cells circumferentially orbit along curved matrix interfaces, then transition toward matrix invasion. And the spheroids are initially slightly elongated, and they round up during orbiting and then embed along the initial elongation axis. And then the mosaic spheroids can orbit and invade with 10% snake cells, but they primarily just invade with 30% snake cells. And the agent-based model successfully reproduced the behaviors of mosaic spheroids. So yeah. And then now I'd like to switch the gear and move on to the mechanical memory of the ovarian cancer. So this was my PhD work. So take the story. So this cancer usually develops on the surface of the ovary with a very heterogeneous stiffness profile ranging from 0 to 100 kilopascal. And considering that healthy ovary is as compliant as 2 kilopascal, this 100 kilopascal to 0 kilopascal regime is very heterogeneous. Just like the other cells, migrations of ovarian cancer cells depend on the substrate stiffness, and they exhibit faster migrations and stronger attractions on steeper substrate. But we should note that the cancer cells leave their home and metastasis. They don't stay on the primary tuber forever. So for the ovarian cancer, they exfoliate into peritoneal fluid, forming multicellular spheroids, and then they float around until they reach the secondary lesion. And we can ask that despite all the challenges, can the cells remember their previous stiffness? And if so, what would be the mechanisms behind it? So to deal with the questions, we established an in vitro model mimicking the peritoneal metastasis of ovarian cancer. So first, we cultured the cells on the substrate with 3 different stiffness levels, which is 1 kilopascal, 11 kilopascal, and 90 kilopascal. And then the primed cells were made into hanging drug culture, mimicking the malignant spheroids in the peritoneal fluid. And then in the end, we quantified how the spheroids embed in 2D and 3D environment to confirm if these cells have mechanical memory they acquired from the first place. Interestingly, the spheroids made with the soft primed cells and then the stiff primed cells had structural differences. Let's see the soft primed cells, and then the nuclei of 1 kilopascal spheroids appears very small, elongated, and they are very crowded. But in the case of the 90 kilopascal spheroids, you can see that the nucleus are more rounded, and then they are more sparsely spaced. And also the actin cytoskeleton structures were really different. For the soft primed spheroids, the actin cytoskeleton form a band here, very dense structure near the surface, but in a steep primed spheroid, there was no such structures. So in 3D reconstructed image, it shows clear differences. Nuclei of 1 kilopascal spheroids are crowded, elongated again, and those in 90 kilopascal, they are more sparsely spaced and round. And then next, we observed cytokeratin 18. Before we jump into the result, so I should explain why keratin 18. So I have some reasons. First, historically, CK18 is a marker for bad prognosis in ovarian cancer, so they express this in primary tumors, CTCs, and cancer stem cells, and so on. And second, this intermediate filament helps actins to reorganize in a stiffness-dependent manner. So if the keratin is knocked down, the actin looks the same regardless of the substrate stiffness. So we assume that this keratin is very important for mechanosensing. And then third, keratin 18 potentially have a significant role in mechanical memory. When the cells are under external force, they make a force-bearing structure which sustains even after the external force is gone. So yeah, we can think that this keratin has a role in structural memory. So here, in the 1 kilopascal spheroids, the CK18 is highly expressed at the periphery, almost wrapping around the outermost nuclei. But in 90 kilopascal spheroids, we can just see through the nuclei. It seems more clear in the reconstructed image. Furthermore, when you block the gap junctions, the structural differences between the spheroids just disappear. So this suggests that the gap junction mediated bit communications are the key link in transferring mechanical memory from 2D to 3D culture. So now we know that they look different, and now we want to know who runs faster. So first, we conducted spreading assays on 2D substrate, and here's the result. The soft primed spheroids were the slowest, while those 90 kilopascal spheroids spread very fast, likely due to their purpura structure differences. And then we embedded the spheroids into 3D collagen gel and measured their 3D invasiveness. As seen before, the 1 kilopascal spheroids migrated the slowest, and notably, 1 kilopascal spheroids exhibit very clear center mass boundaries, even 5 days after embedding, showing the KG structure, the cytoskeletal KG structures are very long-lasting. So in line with this, the growth of the center mass was also biggest in 1 kilopascal spheroid, showing that those cells, those soft prime cells, just chose to stay inside and proliferate rather than go invade outside. So to summarize the second chapter, we found that cells primed on soft substrate develop strong cytoskeletal structures under their surfaces, which are very disadvantageous for 2D or 3D invasion, making the 3D, making the steep primed cells exhibit relatively unregulated invasiveness. So this is the end of my presentation. Thank you very much for your attention, and I'm happy to take any questions.